So our next speaker is Henry Fendel, who is going to be moving us to Rhoda to talk about the AGA. And I will turn it over to you, Henry. I think this works now, and you might need to hold the microphone.
Hey everyone, can you hear me in the back? I'm really excited to present our research on genomic diversity and inbreeding depression in Luta's AGA. Let me pull up presenter notes here. Nope. No, I'm sorry. Oh, here we go. All right. Thank you for your patience. So, hello? All right. We're going to have narration here. This is a test of how good the software is. All right, so before we get into specifics with the AGA population, I want to touch on consequences of small population size. This is going to be really distracting, isn't it? All right. Okay, all right.
The yoga population.
What did it say? I think if we show. Yeah, it just took a second. And then when I started the slideshow, it said something about it.
Subtitle settings. Why is it? I know. Let's just try.
Test, test. All right, I think we're good.
Cool. All right, let's try that again.
Thank you again for your patience. So before we get into specifics with the AGA population, I want to touch on consequences of small population size. Two of the biggest consequences of small population size are increased inbreeding and stronger genetic drift. Inbreeding is just reproduction between closely related individuals. It happens in wild populations. Although many populations do have mechanisms to avoid inbreeding, such as kin recognition or sex bias dispersal, when population size becomes small enough, these mechanisms may not suffice to prevent inbreeding from occurring. Genetic drift, let's think of that as just random chance contributing to which alleles get passed on in the population. And when population size becomes small, genetic drift is going to fix alleles over time, meaning all individuals in the population will ultimately have the same allele at a given locus. Here, natural selection is acting too weakly to maintain alleles that are beneficial to fitness. So inbreeding lowers heterozygosity, genetic drift fixes alleles. A consequence of these two processes is that deleterious recessive alleles may accumulate in the population. These alleles can build up because they're only moderately costly to fitness, and selection is acting weakly. Although any single deleterious recessive allele may not impact fitness strongly, the accumulation of many of them in a genome is actually going to reduce fitness. So once this happens, the question is whether fitness is going to further reduce population size and close this feedback loop, creating an extinction vortex. So we want to know, is inbreeding occurring in the AGA population? And if it is, has it had an impact on fitness? To answer our questions, we sampled 153 crows on Luta between 1993 and 2015. After sequencing their DNA, we made genotypes of all individuals at 1,436 SNPs. Genotypes were used to calculate pairwise kinship and heterozygosity, and then we tested for heterozygosity fitness correlations. I'm going to refer to these tests as HFCs because that's a mouthful. The fitness traits we used in the HFC tests were reproductive success, measured as the number of fledglings produced by a breeding adult for which we had 3 years of breeding data. Survivorship, measured as whether or not an individual banded as a nestling or fledgling survived past 6 months. And then we also used body weight, measured for fledglings only, measured in grams at the time of banding. First, let's get a feel for whether inbreeding is occurring. This matrix is a kinship matrix. It includes all the birds with genotype from Luta. Each row and column represents an individual, and their intersection is the pairwise kinship value. The dark red indicates a very high kinship value, and dark green represents a lower kinship value. So the sample mean here is 0.117, which is on the order of first cousins. In wild populations, this is actually typical. It does not suggest chronic inbreeding has been a problem over a very long period of time. Now, of course, there are some pairs you can see that have very high mean kinship, but again, this matrix includes all of the birds with genotype on Luta. So it's got parent-offspring pairs, it's got sibling pairs, but it does also have 19 breeding pairs. And when we look at the kinship values for those 19 breeding pairs, the mean for those pairs is not different from the population-wide average, or at least our sample average, right? The kinship values for the breeding pairs ranges from 0.073 to 0.32, so that's quite a range. And we do have 2 breeding pairs that we know are more closely related than grandparent, grandchild, or half sibling pairs. So inbreeding is definitely occurring. Furthermore, if we look at our mark resite data, we actually have detected a few pairs that are not in our genomic data set that are also inbreeding. A few years ago, there was a grandparent-grandchild pair, and just this season, there was actually a parent-offspring pair. So again, kinship values, population-wide or sample-wide, not very concerning, but certainly inbreeding is occurring. Here's a map showing individuals at their sampling location colored by observed heterozygosity. Dark red represents 1.5 standard deviations below the sample mean, and dark green is 1.5 standard deviations above the sample mean, right? So sample-wide mean is 0.44. Again, this supports the idea that inbreeding has not been occurring for very long periods of time, and there is substantial genetic diversity remaining in the population. We can also deduce from this map that heterozygosity is fairly evenly spread across the island. There are a few pockets where we have a couple birds that have lower levels of heterozygosity. If we had more data, maybe the area around Rota Resort, the Ibacto area, could be an area of concern, but again, right now, things look pretty good. But let's take a look at how we can investigate impacts of inbreeding on fitness, if there are any. So HFCs, this is a popular means for measuring or assessing inbreeding depression. This is a schematic I've borrowed from Zelkin and colleagues' 2010 review of HFCs. The basic gist here is that we have inbreeding that's ongoing in the population is going to directly affect the markers that make up our genotypes, right? And then we can look for correlations between heterozygosity patterns in these genotypes with a fitness trait. But remember, our genotypes are SNPs. We're assuming that these are non-functional loci. And so we want to be sure that the patterns of heterozygosity in our markers are representative of patterns of heterozygosity in functional loci that actually code for the fitness traits we're measuring. And we're going to do this by measuring identity disequilibrium using the statistic G2. G2 measures the covariance of heterozygosity at pairs of loci in our genotypes caused by inbreeding. The greater G2 is, the more variability there is in inbreeding within the population. So these figures show identity disequilibrium distributions made from bootstraps for each data set. Each data set for a fitness trait contained different birds, so we want to make sure we measure G2 separately for every group of birds. G2 was significant. It was different from 0. Oh boy. In every, for every fitness trait. So the range was 0.13 to 0.19. Sounds like a very small number, but 2014 meta-analysis by Miller and Coltman reported the means value of G2 in 129 published studies is 0.007. Our lowest value for G2 is twice the mean in 129 published studies. All right, so these figures are showing the regression of 6-month survival over heterozygosity on the left, and then fledglings per year over heterozygosity on the right. Neither one of these tests showed any kind of interaction between the two variables, and they don't give us any cause for believing there's inbreeding depression in the aga population. However, the regression of fledgling body weight over heterozygosity is a weak positive correlation, and it is statistically significant with a p-value of 0.0091. So this means that 8.2% of the variance in body weight is explained by heterozygosity. And to be clear, again, similar with the G2 statistic, this is a weak effect with regard to the R-squared value, but if we consider the strength of an effect size that we're expecting when measuring HFCs, this is actually fairly strong. A 2009 meta-analysis by Chapman and colleagues reported that R-squared values from 211 HFC studies range from 0.07% to 3.3%. So we're at 8.2%. Now, total transparency here, all of those studies in the review that I just cited were used microsatellite markers, not SNPs, to measure HFCs. So we do expect a stronger effect size, but this just still gives you the picture that this effect size is significant. Now, given we have detected significant values for identity disequilibrium, we are going to interpret this effect as a signal of inbreeding depression. So the sum of the information I've shared here provides an answer to questions asked many times by those thinking and writing about the aga. Did inbreeding contribute to the decline of the crow? Has inbreeding become a problem since the decline occurred? Because population-wide heterozygosity and pairwise kinship values are relatively normal, it's really unlikely that inbreeding depression drove the decline. But because we're detecting differences in a fitness proxy, body weight, that are explainable by differences in heterozygosity, we do believe inbreeding is now a problem for the population. So the good news is that there's still genetic diversity to be preserved. The ongoing rear and release program, in which roughly 10 pairs of crows, their productivity will be doubled each breeding season, provides an excellent opportunity to target pairs that have high heterozygosity or low pairwise kinship. And furthermore, the release, if areas of low heterozygosity do emerge throughout the island as we add more birds to our genomic data set, releases will offer a great opportunity to infuse new genetic material into those areas that need it most. So yes, it's very concerning we're detecting a signal of inbreeding depression here, but the population has diversity to preserve, and we do have the tools to make a difference. Thank you.
What kind
So yeah, yeah, that's cool. Yeah. The first thing we need to do is add more birds to our genomic data set. We have a lot of blood samples banked that haven't actually been sequenced yet. That's going to give us a better idea of the birds that are currently actively breeding in the population, what kind of genetic diversity that they have. Again, the rear and release project, we are doubling, or we are attempting to double the productivity of specific pairs, and so leveraging information on the heterozygosity or the inbreeding values for those specific birds will help us guide those efforts to further increase genetic diversity in the population rather than like creating more low heterozygosity birds.
Thanks very much, Henry. And we're going to stay on LUDA with the AGA, with a presentation from Sarah Fagri on captive-reared AGA 1 year post-release, survival and behavior. And spoiler alert, there's some cool videos coming up.
Hi, everyone. Can you hear me okay? All right, so today I'm going to give you all a very brief overview of many different aspects of the AGA captive rear and release program on Rhoda, with a focus on the survival and behavior of captive reared birds during their first year post-release. So the first question, why captive rear and release? And Henry touched on this. The primary answer lies in the biology of the species. AGA have a slow life history coupled with relatively low reproductive success, low offspring survival. Fortunately, they have nearly year-round nesting season. Pairs re-nest continuously after failures, and so by collecting the first AGA clutches of each season to rear in captivity, while the wild pairs re-nest, we can essentially double the reproductive output of the pairs we collect eggs from. So the University of Washington and San Diego Zoo have worked together to bring these eggs and hatchlings from wild nests into captivity, where the zoo rears them to adulthood. During 2016, which was our first year of collections, we got 9 viable eggs and chicks. 8 of these survived to release age at 2 years, and they were joined by 2 rehabilitated adult AGA for a group of 10. Here I've got a really brief video just to give you an idea of how we raise these birds. So we go to great lengths to hide ourselves from them. This is like a one-way glass, and then we use a puppet. So they never see our faces. We either wear a costume or put them behind a piece of one-way glass. So fast forward, these birds grew up. Unfortunately, we ended up with 9 females and 1 lucky guy, which limits their potential for pairing up within their cohort. But we released them in an area where they would have access to other wild birds, hopefully wild males. All the birds were radio tagged and monitored daily via telemetry and remote cameras at feeding stations. It's been just over a year since they were released. And here's just a quick picture of our makeshift blind. We had supporters and partners there to view the release. The birds were released. We did it in September and October of last year. We did a soft release. So these birds were held in an aviary at the release site for approximately a month. And then these food hoppers were used to provide supplemental food as they transitioned to life in the wild. These food hoppers, you can see they're also, that perch is a scale. And we have trail cameras looking at the scale, recording video of these birds as they feed, which allows us to track their weight remotely over time. We hoped that these birds would wean from supplemental food in a month or 2. Unfortunately, they were like, free food? Why stop at a month? They ate their free food for almost a year, essentially until we took out the items they liked best. So our initial diet was a mix, similar to what they get in captivity. They didn't really slow down. So the pink and the blue are the male and female weights over time. The bars are the amount of food per bird per day on average that was taken from the supplemental hoppers. And we just weighed this intermittently, which is why there's intermittent bars. So you can see they really didn't decrease their food consumption substantially until we removed all fresh items from the diet. We removed egg and fruit, nothing but dog food and bird pellets less. They were less excited, but even that, they kind of like dog food. We got rid of the dog food and they stopped coming. So who wouldn't choose these juicy items over dog food, right? These are 6 categories of wild food items: lizards, larvae, hermit crabs, adult insects, and fruits. And I'm going to show you a quick video of a bird opening a hermit crab because that's the most unique foraging behavior in the species. So you can see this bird's wedged this giant African snail shell housing a hermit crab. He pecks the sutures to pop the back off the shell, and now he's going to reach in there with his bill and pinch at the abdomen to get the crab to come out. You can see he's keeping his feet well away from this crab. Normally with a large food item, they would hold it down with their foot. He's learned from experience not to do that. Although I can tell you, he didn't learn it permanently. He lost some toes later on. So now he's going to pinch the crab at this joint between the carapace and the abdomen, and he's going to use this really unique, like twisting, shaking motion to separate the abdomen from the crab without getting his feet too close to those claws. There's that twist and shake. If they get it at just the right spot, it separates really cleanly. There, he's got the abdomen free, and then he'll carry that off and eat it. So our main questions in watching the released birds foraging in the wild: are they eating the same things as wild birds, and are they foraging in the same forest strata, the same portions of the canopy? You can see that the released birds did indeed eat all of the same foods, if in slightly different proportions, but this is really positive overall. They ate less insects and more larvae than their wild counterparts, more lizards, but we do see them feeding on all the same foods and a good success rate. In terms of forest strata, we also see them using everything from the ground to the super canopy, just as they should. There are some differences. Our released birds tended to forage more in the lower third of the canopy and less in the upper third of the canopy than their wild counterparts, but luckily they didn't forage too much more on the ground than their wild counterparts. That was a concern. When we rear them in captivity, we try to encourage them to use the upper areas of the aviary, although we can never imitate, a true forest canopy. Social behavior is another big question. Are they interacting normally with their wild counterparts, with each other? And group size. Group size is really interesting because historically aga did form flocks, especially non-breeding individuals. And there's some benefits to group living that the aga on Rhoda now, because they're so dispersed and few and far between, subadults, non-breeding adults don't really have the opportunity to form flocks. We hope that by releasing a cohort of 10 birds, we could recreate this opportunity for group living. You can see that the released birds did indeed spend more time in groups, 4 or 5 birds, which may not sound like a big group, but generally when we see wild birds, it's just 1 or 2, like a family group or a single individual. Spatial behavior. So this is our release site. This big blue square is where we released our birds. The green circles are territorial wild pairs. And you can see they used a really large area. They fit themselves in nicely within existing habitat between the wild birds. Our red triangles are nesting. So we had a number of nests. That one male was pretty busy. And I've got a table to show home range sizes. So this is comparing their home range sizes with those of wild birds. Our released nesting pair, you can see I've got an N of 2 because it's just these two birds, had an average of 35 hectare home range, which is similar to the 53 hectare home range we see for wild paired adults. The subadults used much, much larger areas, and the unpaired single ladies in our released cohort used an equivalent amount of space, 200, 275. This is just another graph. This is looking at home range area over days post-release. And you can see that they began exploring right away. They at least explored a perimeter of this whole area within 60 to 90 days. And beyond that, they continued going back to different areas within the larger area they'd already explored. Survival and nesting, the two most critical aspects of judging our success. So 3 of the released females died during the first year. 1 died at about 6 weeks. The other 2 died around 10 months post-dispersal. This survival rate is equivalent to first-year survival of AGA, and we were quite happy with this survival rate. Cause of death? Well, the only known cause of death among these 3 is something that is called AGA eukaryote X, or the mystery disease, with 3 question marks to signify that there are more questions than answers about this disease. I could go into it, but we've got these cells that are believed to be a foreign agent, yet despite years of intensive investigation, it can't be identified. So most of these birds are not dropping dead of a mysterious disease. Mostly they're spending their time foraging and nesting. Here is our male collecting nest material from a coconut. This was about a month post-release, maybe less. So really we see these birds get right to it. On the day of release, they're already foraging for wild foods successfully. A few weeks later, they begin nesting. And here is their nest. You can see they built a beautiful nest structure, laid normal-looking eggs. Unfortunately, their eggs did fail to hatch, but it's a great start. So, summary and next steps. The survival and behavior of these first 10 birds paint a really positive picture of the potential impact of the program. Determining causes of mortality is important. Determining the identity of this AEX, this inflammatory syndrome, which is causing both wild and captive reared birds who are seemingly vibrantly healthy to just suddenly drop dead. Nesting success is going to probably be the even more critical aspect of evaluating our program's success in future years. 70% survival is great. If all of those birds can pair up and nest successfully, we're going to be golden. And we've got an upcoming release later this month. We've got 13 2-year-olds that will be released at the same location. Hopefully some of those single ladies will get lucky, because this time we've got 7 males joining the population to round things out. With that, I'd like to thank our supporters, funders, partners, the huge number of staff, students, and volunteers that have made this program possible. And I'll take questions.
Hey, Sarah. Have you observed this AEX, you said, in wild birds as well?
Yeah, it's been the cause of death in wild birds during the past, I'd say, 5 years. Every mortality that we send in has this signature of inflammation, systemic inflammation, but primarily in the lungs and liver. So these birds are dying of respiratory failure and liver disease. We only saw it for the first time in a captive reared bird just this past July, but it's something that's been investigated in wild birds for a long
Like
time.
big numbers in the wild birds, or?
The numbers that have died?
Yeah.
I mean, we've sent in probably about 25 carcasses with these lesions.
Okay, thanks.
Yeah. Yeah, so the question was if the wild aga pairs exclude the released birds from their territories. If they have an active nest or a fledgling, they'll exclude all other individuals, wild or released, from within a couple hundred meters, but they don't exclude them from their entire home range. It's a pretty small area, and it's only during active nesting, so there's really very little space that they're excluded from.
Let's thank Sarah again. And we'll move into our last talk before break about Mariana Common Moorhen population census and habitat modeling with Steve Mullen and Brad Eichelberger.
Good morning. Okay, so we're going to do something a little bit different here. We're going to tag team this particular discussion. My name's Steve Mullen, Brad Eichelberger. We'll be talking about the Mariana Common Moorhen Population Census and Habitat Modeling Project. And we're at DFW. So I'm going to start off by talking about the actual bird surveys and kind of discuss some of the results we found with that. And then Brad will take over with the habitat modeling, mostly because he doesn't trust me discussing his modeling techniques. All right, I have no idea what I'm doing there. Thanks. Okay, so in 1984, the common moorhen was listed as endangered. It exists currently only on the islands of Saipan, Tinian, Rota, and Guam. There used to be a population up in Pagan, but that population since has been extirpated. The reason it was listed is there was an observed population decline, as well as some known limitations to the populations. So I'm going to just talk about those limitations real quickly. So habitat loss being one of the main ones through development of wetlands, as well as the encroachment of an invasive species of Phragmites. So the Phragmites was encroaching onto the wetlands, removing or decreasing the amount of the area that the moorhens were able to use. Some of the other habitat, or some of the other limitations are nest predation, so rats, down in Guam, the brown tree snake, and other human-induced fatalities like automobile strikes as birds are trying to cross roads. Since I don't have my clicker, I forgot to change the slide. So here's a picture of frag encroaching in on one of the wetlands here on Saipan. I do want to make a comment here that, yeah, there are all these limitations in place, but the species does at least appear to be somewhat adaptive, somewhat flexible. Here's a moorhen nesting on a garbage bag up in the Marby landfill. So this was only taken a couple of years ago, a couple of days ago. So I know a few people here in the room have already seen this bird nesting up there, but I thought it was a good place to talk about the flexibility of this species as well. A complete Marianas-wide survey hasn't been conducted since way back in 2001, so it's a long time ago. It's about 20 years ago. So DFW was really keen on getting out there and trying to figure out how this population was doing. And that was one of the main objectives of this project. So the survey sites that we checked at, so I'm going to take a step back here. So we were working hand in hand with Guam DAR, and we were scheduling all the surveys together. Guam DAR was taking care of Guam. They were doing the surveys down there, and we were scheduling them so they were all being done at the same time. So I'm only going to be discussing really the CNMI survey sites. Saipan, we had 45 sites spread across the entire island, so we hit pretty much every single wetland available here. Then we had an additional 2 on Tinian, and then the various golf course ponds and wastewater ponds over on Rhoda. So before I forget, I want to just throw a thanks out there to Nathan Johnson, who's here with Micronesian Environmental Services. Him and Noah Burrell with NAVFAC, these guys helped out with the Tinian sites. And I'll forget if I don't think of them now. So we completed 3 sets of surveys. We did 2 in the dry season, 1 in the wet season back in, well, 2018 and 2019. They were all conducted by foot or by kayak. The length of each particular survey was determined by the size of each individual wetland. So they range anywhere between 10 minutes to almost 2 hours. And you can see if you go out to a golf course pond that doesn't have a whole lot of vegetation surrounding, you can get that done in 10 minutes easily. But you go over to a much larger wetland like Sasupi Lake, you're out there in a kayak, it's going to take much longer. So again, the type of methods we used was dependent on the wetland also. So if line of sight was obscured by vegetation or Phragmites, most likely, we had to use point count. So we had to listen for the birds. There was no way we were going to see them. When open water was viewable, we used direct counts, of course, and sometimes we had to use a combination of these 2 methods. So here in the CNMI, we had an average of 161 adult moorhens across these 3 sets of surveys, along with 23 juveniles and 5 chicks. Interestingly, on Saipan, every single set of survey, 50% of the wetland sites were occupied. This was independent of the season, so wetland or wet season, dry season, it didn't matter. What did change was the individual wetlands that were occupied, but every time it was interesting enough that only 50% were occupied. In May 2019, this year, was very dry. We found that 72% of the adults were using only man-made wetlands, so golf course ponds, the big Japanese water tank over in Puerto Rico. So these are the sites that we're using. The ephemeral wetlands were drying up, the natural wetlands were drying up also, and then frag was taking out the rest of the usable areas. And this just really cemented one of the ideas that we've had for a long period of time, that these man-made wetlands are incredibly important to the CNMI during these dry seasons. Those ephemeral wetlands are no longer available to them. And then, of course, we pretty much knew that we'd have a higher number of detections during the dry seasons. Again, these birds are flocking to areas of open water, areas that water's available to them all the time, with fewer limitations to observation. Next, we wanted to compare what we found to 2001. So in 2001, they used the survey where they had the greatest number of bird detections. So that's what we did, so we could compare the 2 numbers. Back in 2001, they found 195 adults. 2019, we had 190. So, big question is what's going on with the population? Is it stable? It's kind of difficult to answer that question with only 2 data points. At least it looks stable. It's not decreasing as far as we can tell. So, kind of wanted to add as much data as we could to this. So, we wanted to draw upon other surveys that are conducted here in the CNMI. So DFW runs biannual waterbird surveys here. It's on a subset of the common moorhen survey sites. But we took this data and took a look at this, and at least over the short term, the past several years, Saipan looks to have a pretty stable population. If you look at Rhoda, it almost looks like a small increase in population there. It's not a large number of birds anyways, but again, we just wanted to kind of have a little bit more data to fall back on and at least get an idea of what these populations are doing. And using the best available data, it does appear that it's a fairly stable population. And, that's pretty much the best conclusion we can come with what we have. And so I'm going to pass it on to Brad now to continue.
Thank you, Steve. So another aspect of this project was actually looking at predictors of distribution. So this had been done before in 1999 on Guam, but this is the only study that's actually looked at moorhens, looking at spatial distributions and things along predicting occupancy and things along those lines. So basically, in 1999, Ritter and Savage used logistic regression, and they looked at habitat composition of the wetlands using aerial photography, mapping out the percent area that's covered by each habitat type, along with things along the lines of wetland area and also disturbance variables. So looking at distance to road, distance to buildings, and things along those lines. What they had found was the only predictors were wetland area and emergent cover were actually negative predictors of presence-absence for the moorhens on Guam. So basically, smaller wetlands with less emergent habitat cover was actually better predictors of where moorhens would occur. But they actually didn't think that was a very good model, and they concluded that it's most likely that the wetland availability is the primary factor determining where these species go. So we wanted to do something similar. So we repeated using the same modeling technique, which is stepwise logistic regression. But also, times have kind of changed since 1999. There's a lot of more machine learning models out there, so we incorporated boosted regression trees as well. And this is, just to be clear, this is only using the data from Saipan for this. So we included detection variables that Steve had from his surveys, so things like time of the survey, or length of the survey, noise variables, percent cloud cover, and also rain categories and things along those lines. And also looked at habitat composition using satellite imagery. So we looked at the area of open water, Phragmites, emergent, and shrubland wetland habitat. And this is actually pretty cool because we could use, like I said, this world-view satellite imagery. And this is 40 to 50 centimeter resolution. And it's, we're basically able to look across the 3 sampling periods. And the really cool thing about this is we're able to get within plus or minus 3 days of each of the survey dates that Steve went out. So just to show a kind of quick time lapse, this is hopefully going to work. So this first image that we're looking at is taken in June 3rd, 2018. This is actually during the survey period for Lake Susupe. And we're going to go through here, and the next image you're going to see is the wet season. So this is the dry season, 2018. This is the wet season, 2018, pretty much the same. But this is the dry season, 2019. So you just kind of flipping back and forth, you can really see there's a lot of water drawdown. It was definitely a huge difference in terms of the water regime for these sampling periods. So looking at the results, and I got 3 minutes to rush through this. So looking at the results, this is the logistic regression, and if we actually use the dry season 2018 as the training data set, it kind of gives us a really unique opportunity to use the wet season 2018 and the dry season 2019 as kind of unique variables for validating the model, because we can look at within the same year of 2018, different seasons but same year, and also look at same season but different years. And what we kind of see is that the only predictors that came out were time in terms of minutes and the amount of open water in square meters, and more so that the longer the survey is, the more likely the chance that you're going to find a moorhen, which is kind of common sense. But we already know that the time's actually correlated with the area of the wetland. When we start looking at the validation statistics, I don't want to really get into this too much, but basically, as we test the model with the other data sets, the further we go along throughout the year and towards the dry season of 2019, the less likely we are at actually capturing sensitivity, which is actually the ability to correctly categorize where a moorhen's going to be. So this is kind of important. It says that in the dry season of 2019, the model doesn't quite fit. Same for the boosted regression tree. The two variables that come out most importantly are going to be time and open water as well. And once again, we see, at least down here, that our predictive validation metrics actually decrease as we go towards the dry season of 2019. So it makes sense, like, why don't we just build a model with the dry season of 2019 and see what happens? Well, nothing, because there's no model convergence for either logistic regression or boosted regression trees for that data set, which means the variables that we're measuring could not predict anything in terms of presence and absence. So in conclusion, as Steve had talked about, the population appears relatively stable, and basically these man-made artificial wetland sites are pretty crucial during the dry season as they provide water resource and habitat refugia. And based on what we're looking at, the predictors are not well identified. Some sites have changes in water regime, while others kind of have permanent water. There's obviously shifts in occupancy between the different sites. So our thinking is maybe in the future we start looking at these water regime variables, if we could put data collectors in there to look at drawdown and things along the terms of how much water is in these wetlands. Another possibility is we're actually getting bathymetry data into the CMI, so we could basically backcast the depth of the water using the satellite imagery and the bathymetry and kind of get a better idea of how much water these wetlands are holding, and then incorporate some weather station information to see how long it's been raining and how long these things are holding water and things along those lines. So with that, a couple quick thank yous, and I'll open it up for questions.
One quick question before the break. Anyone? Back there?
This isn't a question just for you, but that was great on the Palatat. Just two comments. One being, if it would be really great if we could see more use of the Chamorro names of these birds, the moorhen being Palatat, the swiftlet being Zazaguac, and just incorporating that would be really nice. As a local person, I would really appreciate that. And another thing is that I would like to know, with all across bird species, is there anyone doing work looking at their resiliency through storms? Since we have so many storms coming through the area, what are the bird responses? How do they prepare? How do they live these things out? And what can we do to be assisting them?
I believe Lauren's going to be talking about that soon, but actually I want to also add that this year I have a project that's going to be looking at satellite imagery and looking at typhoon response, or sorry, vegetation response and resiliency in terms of typhoon events over probably the last 5 years, and adding in different bird populations, trends lines, and things like that on top of it as well. But excellent question.
Thank you. Don't get up yet. One quick announcement from Jill, and then it's time for a break.
Okay, welcome to the folks. I saw the flush of folks come in that came in on the United flight. So just very quickly about the field trips. Folks that got a lucky seat on the boat to Managaha today, you're going to have to leave the conference early today. For any of the field trips, if you don't know where you're going, ask me or ask anyone from Saipan. These are easy locations to find. There are spots available on the Wednesday morning field trips that you can sign up back at the registration desk. And also, if you signed up for a trip and you find that you're not able to go, I'd appreciate if you would check in as well and let folks know about that. So, so hit the registration desk for field trip information and enjoy the refreshments, and we'll be back in a few minutes.